So I would like to thank everyone for being here. I know it's late in the day, and you must be exhausted with all the information. So it's a different company, so I'm going to go through this, and I'll take questions at the end. I think we have some room in the back. So Cesca Therapeutics actually is a new name to a company which was known as Thermogenesis. Thermogenesis is being a public company, 30 years in US. So 3 years ago, Thermogenesis, originally a manufacturing device, a medical device manufacturing company, merged with a company called Totipotent RX, which was a clinical company using Thermogenesis devices. So when these two companies merged, it became Seska. So I'm going to talk more about this and what do we do. Being a public company, I may be making some forward-looking statements. So this is the slide to cover me up. So who we are, Cesca Therapeutics. So Seska is a leading regenerative medicine company that develops, commercializes, and markets automated technologies. So we have a long history and expertise of developing automated systems. Yesterday, I remember one of the speakers in the panel mentioned that most of the technologies we are using came from blood banking or bone marrow processing systems. So Thermogenesis, which is the device division of Seska, is basically the first company who developed the automated processing AXP system for cord blood biobanking. We are very proud of it, and so date we have processed almost 1 million samples in the cord blood industry. So basically we have two divisions. One is the device division, which is Thermogenesis. So we have developed and we are continually developing automated systems for clinical biobanking, point-of-care applications, and automation. And we are in development for automation of immuno-oncology applications. On the therapeutic division, we are using these technologies, and this activity started 7-8 years ago with Totipotent. So they were using the platform technology developed by Thermogenesis to develop autologous bone marrow concentrate-based therapies to address significant unmet need in vascular and orthopedic markets. This is a snapshot of the vital stats of the company. Just the highlight here. So basically recently in November of 2016, we have a major investor from China called Boya Life Group, and they are 70% owner of the company. So Seska, so this is what I'm talking about. So Seska basically is a clinical development company with the IDE, phase 3 IDE trial in CLI in US. We have 2 branches, device division, well established. We generate revenue from this. And the clinical development company in India, which is called Totipotent RX. So the basic model is we do early stage human feasibility studies in India, cheaper, faster, and depending on the results of those studies, we take interesting indications to next stage of development, like we did with the critical limb ischemia. We bring it to US and take it to FDA for the next stage of the trial. So this is just to show how extensive we are in the device company. So all major blood centers inside US, outside US use our technology, which includes automated cryopreservation system. This is called bioarchive, along with the processing technologies. This is the basic overview of the technology. So basically, in terms of cell therapy, most of the companies consider this as a primitive processing technology. However, the device we have is different from the available devices, physical devices in the market. So we use algorithm-based device, a smart vision-based algorithm for separation of cells. Is still the Buffy code, the stem cell fraction, but we can actually modulate the amount of RBC and granulocytes, and we can change the composition of the product using the same device. And you do not have to take it 5 times out of centrifuge. It's a single centrifugation which can be programmed, totally hands-free operation. And unlike other devices, which mostly talk about viability or recovery only, we do a complete range of analysis, which not includes viability and recovery, but also the potency, which in terms of CFUs, the migration assays, the tube formation assays. So we check at each step of our devices and systems that we maintain the bioactivity as well as the viability and recovery. So these are the 3 branches. For cell-based therapeutics, we have. This is commercially, the first two are commercially available. The third one is in development. So basically, we do clinical biobanking. So we provide AXP with automated bioarchive for corporate banking. It's been used worldwide. We have a system called MXP, which could be used for processing of bone marrow at the surgical sites. We have clearance for this. And this one is, I'm going to talk at the end of the slide, is basically we are developing an automated manufacturing expansion and storage technology for cell therapies like T-cell or natural killer cell-based therapies. So basically, the strategic focus of company is in the three fields. First of all, advancing our proprietary technologies for automated point-of-care processing. So what we do in here, so our devices could be used, and we have developed a system where the bone marrow could be harvested, processed, even quality checked, and delivered back to the patient within 60 minutes. So most of the devices talk about the processing, crude processing, but we have this automated processing system, and we also do the quality check to ensure that a minimum threshold dose are delivered. I can talk about more, but I have more slides to go. The other thing what we are doing is developing clinical applications around this automated point-of-care platform. So these are the focus for Seska side. In cardiovascular, we have critical limb ischemia, which is a late stage of peripheral arterial disease, acute myocardial infarction. We have done one patient proof of concept study. In orthopedic indication, we are focusing on AVN, avascular necrosis, OA. We have talked about a lot in this conference, and non-union fractures. So this is our lead indication, critical limb ischemia. Sorry for the bad pictures, but thank God it's after lunch. So it's more than 1 million incidents in US. So the population we want to target is basically the patients who do not have any option other than amputation. So in US alone, we have 70,000 major amputation above the ankle, and 134 minor amputation. So in total of exceeding 200,000 amputation in the US. These patients are at the end stage of the disease, and they do not have any other option. I know in this conference, bone marrow concentrate-based therapy are still considered primitive, but this therapy has been in field for a long time, and this is one of the recent meta-analysis published by Regenerative et al. In Circulation Research. In this, just to just to shed some light on the literature. So basically, they summarized all the trials available till 2016, and they took 19 randomized controlled trials with 837 patients. They excluded small studies. The studies have high heterogeneity and publication bias, but even with that limitations, the authors concluded the primary analysis, which included only the randomized controlled trials, that the cell therapy, even this bone marrow-based autologous cell therapy, reduced the risk of amputation by 37%, and it improved amputation-free survival by 18%. Wound healing is more effective at 59%. So basically, if you want to summarize, this therapy for this patient population has a potential to avoid 1 amputation per year for every 2 patients successfully treated. So we have done 2 feasibility studies, one in Italy, one in India. The Indian one is intramuscular studies, and literature shows like intramuscular is better one. This is the result for the intramuscular study. We treated 17 no-option CLI patients, who were recommended for leg amputation. This is the 12-month follow-up data. I'll just highlight some of the points. So we got major amputation-free rate of 82.4% in this no-option cohort. What's more interesting is basically we did the CTA study, and we found both statistically significant collateral vessel number and size in distal thigh and proximal leg. So basically very interesting data. We took the study data for both the studies and some more experiments, and went to FDA. So to get a IDE approval. So we have a US FDA phase 3 IDE approval, and this is the study design. It's a 362 patient, Rutherford category 4 and 5, no and poor option subjects. We have up to 40 clinical trial sites allowed. the primary endpoint is difference in amputation-free survival over 12 months. So this is one of the indications we are looking for a strategic partner. Phase 3 IDE studies are not cheap. We can do the feasibility studies. But for this one, we need the money, as well as we're looking for a partner who has more expertise and also more scientific and clinical power to take this program forward with us. This is another indication I did not mention on my slide, non-healing ulcers. So we did a platelet-rich plasma-based application for non-healing ulcers. We recently published it. You can read the paper. But in summary, if I just, these are the pictures. So picture speaks 1,000 words. So this is just the summary of. So you can see we saw very good results in wound healing. And we have wounds of different etiologies. Diabetic wound was the major one, proportion of it. So, and to summarize, we saw basically there were no adverse event. It's autologous therapy, so adverse events are not there. The one thing we observed, in some of the patients, we observed wound healing as early as 4 weeks, and the mean healing time for all the patients were like 8.2 weeks with plus minus 1.9. So this is a snapshot of the pipelines we have. So we have data on all the orthopedic study, which we haven't shown, but if you need, I can talk more about it offline. The third one is, this is the development stage. So we are in process of developing automation for immuno-oncology application. So our vision is basically having a system called CXP system, which will provide end-to-end solution from the harvest to the cryopreservation and also allowing a physician to get cell therapy at the pharmacy. So basically, and the CliniArchive, which is the next version of the bioarchive, would help us do it. So basically, if you want to get a vaccine, you can take an analogy of a vaccine. So vaccines are developed a long time ago, and now if you need a vaccine, you just go to a doctor, he orders the vaccine, the nurse picks it out from a refrigerator and gives to you. Cell therapy is there. So cell therapy is ready, or maybe in 5 years, it will have the same thing. So there will be so many cell therapies in the market, a doctor would have option to use any one of them. But the major issue that we see is the cold chain management, the supply chain management. So every time a physician needs a cell therapy, he has to go back to the manufacturer or the core storage lab to say, I want it, and he has to maintain a cold chain processing and schedule the patient so he gets the viable therapy in time. What we see is, like with our CliniArchive, which has a capacity of storing more than 3,600 samples in liquid nitrogen phase, we can put that in pharmacy, and the beauty of the system is none of the samples touch each other, and they can be handled independently out the system. So you can store as many cell therapy as you want at the pharmacy, and it can be ordered at the discretion of physician. So that's the future, we think. So this is just the last slide. I will go into it. So basically, most of you know about the immuno-oncology cell manufacturing. So it starts with the source, which could be bone marrow, peripheral blood, or mobilized peripheral blood, or leukapheresis. It goes to the lab. You do isolation of MNCs using manual methods such as FICOL. Then selection of cells. It could be CD3, gene modification, or it could be without gene modification, expansion, volume reduction. And then the cells are finally put into a formulation for infusion either directly to the patient on the same day or cryopreserved and transferred to the clinic for infusion. So basically, Seska has, or Thermogenesis has developed technologies for some of the points here. What we are taking forward is basically filling up the gaps of some of the points we have, such as selection of cells using our technologies, volume reduction, because when you do a cell expansion, the volumes are much higher. So we are developing technologies and putting an integrated system which can be used by any lab without the need for a GMP core lab to process this and do this in a remote area or less advanced area. So these are the milestones for the company. So you can read it. So development of the immuno-oncology CXP system is one of our major focus, and to secure a strategic partnership for our clinical programs. And with that, I'll thank you all.